Hello everyone. In this video I'm going to be talking about something called meiosis, and I'm going to include in it a little bit about genetic variation and why meiosis creates genetic variation. Now, in another video entitled Mitosis, I talked about cell division of body cells. This meiosis is different because this is cell division that creates not body cells but instead the gametes, and by gametes I mean sperm and egg. The whole purpose of meiosis is not only to create cells that have half the genetic information, so that when one sperm meets one egg, you can come together and get a full set, but it's also to provide some element of genetic variation so that the sperm and egg are slightly different. So we're going to outline the steps of meiosis, how you go from one cell to actually creating 4 non-identical cells. And then we'll look at why or how variation comes about. So let's start first of all with one typical cell here. We're going to draw in a nuclear envelope, and like in the mitosis video, I'm going to draw one set of chromosomes. There are 46 chromosomes in 23 pairs in every cell. And I'm not going to draw all 46. Instead, I'm just going to pick one random pair, and we're going to draw in red the maternal and in blue the paternal. So maternal means from father, P for paternal, M meaning maternal from mother, and the P paternal meaning from father. So we're going to just draw in one pair of chromosomes in that nucleus in a cell. Now, we have, like we had in mitosis, a period called interphase. Now, in interphase, we have growth of the cell and the formation of all the proteins and all the different organelles found within that cell, but we also have a phase called the S phase, and that's a period of replication, because we do need to copy our DNA. So we get copies or replicates of our chromosomes. So in interphase, what we're going to end up with ultimately, just draw our nuclear envelope in place again. We're going to have our red maternal chromosome and a copy. We're going to have our blue paternal chromosome and its copy. Now we actually say, because this maternal chromosome, the one I've labelled here, went with this paternal chromosome, or naturally would go with that, we call that a homologous pair. If I just draw at the top here, a homologous pair of chromosomes are ones where you have the same genes, if you like, on both at the same position. So if we had a gene here so in this diagram at the top, I'm just imagining we've drawn a random pair of chromosomes. If we had a gene here for the ability to tongue roll, on the opposite chromosome we'd have an equivalent. So this would be maternal, this would be paternal. We have the equivalent. Now, I've said in a previous video that we refer to one as gene, and an alternate version of a gene would be an allele. So homologous pairs of chromosomes are ones where we have alleles for all the different genes, but they're matching, they're in the same position along the chromosome, or the same locus, if you like. They go together. That's what we mean by homologous pair. And that's what we're dealing with in interphase at the moment here. This pair is a homologous pair. So that maternal and paternal chromosome go together. So now we've had interphase and we've replicated all our DNA. We have a period called prophase. More specifically, prophase 1. Because unlike mitosis, so cell division of body cells, we're going to have phases occur again because we have 2 distinct periods of cell division, if you like. So for the moment, we're going to look at prophase 1. Now, in prophase 1, we have that cell and nuclear envelope actually starts to disintegrate, and what we get is a shortening or fattening of our chromosomes. They condense, they become shorter, and they become more visible as actual strands, so a bit like this, if you like. So I'll draw them, then I'll explain what the little dot represents. And I'll draw it in here, but this nuclear envelope will start to disintegrate at the end of prophase 1. Now, this dot that I've drawn between the two, so if I take just out of here, if I just draw it separately, what I've drawn here we call the centromere. And the centromere is the point of attachment between one chromosome and its replicated copy. So we can see in the original picture that I drew in this video, we have one red, one blue, and then the red chromosome copied, the blue chromosome copied, and those two copies are attached to one another at this point here, at this central centromere, which is what the two big dots are. Now, from now on, we're going to refer to each chromosome, if you like, and its replicated copy as a chromatid, or more specifically, sister chromatids. So like I did in the mitosis video, this red chromosome here is one chromatid, and this one here is going to be its sister chromatid. We can take the strands as original, or as individual rather, entities from now, even though they are joined, because we will be referring to them as separate strands shortly. So in prophase 1, just to reiterate, we have the cell with the nuclear envelope that at the end of prophase 1 will disintegrate, and our DNA condenses, it shortens, and it becomes smaller, and we can see it as two individual strands on each chromosome, essentially its original and its copy, which we're going to refer to as sister chromatids. Also in prophase 1, proteins called centrioles start to form either side of the cell. So then from prophase 1 we move into what's called Metaphase 1. And in Metaphase 1, just like in mitosis, we have our cell here, nuclear envelope's now disintegrated. We have our centrioles ready either side, here these little proteins. And we have lining up along the equator our homologous pair. So we have the maternal chromosome and its copy, or the sister chromatids. We have the paternal one and its copy. So this homologous pair of chromosomes line up along the equator in the middle of the cell. Now this is where Metaphase 1 then leads into something very different than just metaphase in mitosis. In mitosis, in normal body cell division, the spindle, these protein fibres here, the centrioles that go to form what's called a spindle, would typically grab, if you like, one of each pair in the chromosomes. So in mitosis, you'd have one red chromosome, if you like, and one blue chromosome going left, and you'd have one red and one blue going right. But something different happens here. In meiosis, this centriole will produce a spindle fibre, a protein spindle fibre, that will essentially grab the entire chromosome and its copy. So the entire of this red structure that we see, the chromosome and its copy, this spindle will grab, and this one on the right hand side will join at the centromere to grab the blue chromosome and its copy. Those two blue sister chromatids together. So rather than splitting apart sister chromatids like we have in mitosis, meiosis will grab the entire structure. Now metaphase leads on to a period called Anaphase. And this again would be Anaphase 1. Now anaphase is when the spindles start to contract and they pull the chromosomes to opposite ends of the cell. So if we imagine that happening, what we'll have is something like this. So we put our centrioles in. We'll have the red chromosomes going one way. If you imagine, so the red one's going this way and the blue one's going that way, you can see that what we're going to do is take two chromosomes, if you like, to opposite poles of the cell. So if I just shrink this down, we shrink this down, put it into the corner here, just to create a bit more room, and explain the significance of what's just happened. After anaphase, we have a phase called Telophase 1. And that is when the cell starts to split in two. And it splits along this line here. So we get what's called an invagination. It starts to come in essentially from these ends here, from the middle. And we're going to imagine these two cells. If you imagine those two red arrows meeting, we're going to split this cell, one cell into two, along that line. Now, if you see on the left-hand side, we have one red chromosome and its copy, and on the right, we have the blue and its copy. So we've kept them intact. Rather than separating individual chromatids, we've kept the original and its copy together. Now, this is called telophase 1 because we split these cells, and again, like mitosis, there is a distinct phase called cytokinesis, which is the last point at which these cells divide. So the very last point at which these two cells split, we get a phase called cytokinesis. But these two cells here, when they're formed, would look a lot like this. Now, I said the purpose of meiosis was to create cells that had half the DNA. Now, if you look at the original cell I drew in this picture, we started with two, one red, one blue, two chromosomes. In these cells that I have at the bottom now, we still in a way have two chromosomes, albeit the original and their copy. So we have another distinct phase of division. We go through what's called prophase 2. And I'm going to draw all of these on the same picture on the bottom. So in prophase 2, two new spindles, or centrioles rather, will form in each cell. And they are referred to as spindles. And they'll each produce spindle fibres. And these spindle fibres will attach now one to each chromatid in the pair. Now if you imagine pulling those apart, so after prophase 2 we again have a period of, well, we would have metaphase 2, so I've only drawn one pair but there are 23 pairs in the cells, so if you imagine all 23 lining up in the middle in metaphase, they'll then get pulled apart in anaphase 2. So if I again further shrink this down, if we take this, if we take this cell here, we can see what we're going to get is something that looks a bit like this. We're going to have the spindle fibres pulling one sister chromatid one way and one another. And the same thing would happen in this cell here, we'll have those centrioles with the spindle fibres pulling one of the blue chromatids to one side and one pulling one to another. Now, if you imagine that happening, so this would be Anaphase 2. If you imagine both those sister chromatids with the red and the blue, so the red cell and the blue cell essentially, if you take the red sister chromatids and pull them apart, the blue sister chromatids pull them apart, then imagining having Telophase 2, so those cells splitting, what you'd ultimately end up with, if we just draw a big arrow to show our end point, what you'd ultimately end up with would be 4 cells. So in one cell you'd have one sister chromatid, which we can now recall a chromosome, and we'll put the nuclear envelope back round because it would reform. You can see you'd also have another cell that would have that sister chromatid in, and the bottom two would have a nuclear envelope forming, and we'll have each of those blue chromosomes. So essentially what I've drawn are the 4 cells you get when this cell here would ultimately split into 2, and the 2 cells you get when this cell here would ultimately in telophase split into 2. So we can see we've created 4 cells at the end. Now each one of those cells has 1 chromosome, and in the original cell at the top we had 2. So we have created 4 cells. So we'll just summarise it here. We've created 4 cells. Each of those 4 cells has half the DNA. So because they have half the DNA, we call them haploid. See, whereas mitosis produces diploid cells with a full set of chromosomes, we produce 4 cells that have haploid. They have half the original amount of DNA. So I said at the beginning that there are 46 chromosomes per cell. So meiosis would produce sperm and egg with 23 chromosomes. It's just in this example, for simplicity, I drew 2 chromosomes at the beginning, one with an M and P, one mother, father. And now we've got 4 cells that each have just 1 chromosome rather than 2 in. So we have 4 cells that are haploid. But if you look, you'll notice that in the 4 cells, there appear to be 2 that are red and 2 that are blue. And I said at the beginning of this video that meiosis creates 4 non-identical cells. So I've not drawn it here, I've just outlined the basic method of meiosis and how you go from 1 cell to get 4. But clearly there must be something going on in this process to allow for genetic variation or differences between those 4 cells. So we're going to finish this video by outlining the 2 places in this whole process where you get this genetic variation coming about. So I've shrunk the screen a little bit, and I'm going to circle in green at the 2 stages where we get this variation. Now one happens during prophase 1 and one at metaphase 1. So we're going to talk about each one in turn. Now in prophase, so in prophase 1, what we had was, and if we just draw it a bit larger here, and I'm going to, in fact, use colours to represent this. Here was our maternal chromosome, and here was our paternal chromosome, our father's, or one that came from father. And I said that each one would have made a copy. So during interphase, they would make a copy of each other. So, do a copy here. And this one will colour in red. And again, we have a copy of the paternal chromosome, and this one again will colour in blue. Now, during prophase, what happens is something called crossing over. So you get this process of crossing over, and a portion of one of the sister chromatids, because if you remember we said that when you have the chromosome in its copy, we then refer to both as sister chromatids. So this portion here can actually exchange with this portion here. They cross over. The point at which they cross over is called the chiasmata. That's C-H-I A-S-M-A-T-A. Chiasmata only comes up at A level and IB. And they almost swap chunks of DNA. They swap parts of genetic material. So if you imagine the bottom chunk of one red chromatid swapping with a chunk of the blue chromatid, what you'll end up with is something that looks a bit like this. You'll have one red chromosome or chromatid here, and then you'll have its copy that would be red with a portion that has changed and is now blue. And on the paternal side, you'll have this blue chromatid from the father with a new portion that is now red, and then you'll have a whole blue one here. Now, if you think about what you make at the end of meiosis, where you get 4 non-identical cells, you can see now where this difference comes from. Because if we were to take these 4 chromatids that I've drawn, the maternal and its copy and the paternal and its copy, we've now actually created, by crossing over, 4 very distinct different strands of DNA. We have one that is red, one red with a bit of blue, one blue with a bit of red, and one that's just blue. So if we go back to this stage here, you can see that in real life you wouldn't have 2 red and 2 blue. In real life, one would be red with a little bit of blue, one would be blue with a bit of red, and one would be fully blue. I'm just using the colours for visual purposes in this video. But crossing over explains why those 4 cells that are all haploid are genetically different, and that's key. Those 4 haploid cells are genetically different because of crossing over, but also because it's something that happens in metaphase 1. So in metaphase, when those homologous pairs line up along the middle of the cell in the equator, it's the order that they line up on is really important. In mitosis, it doesn't matter, but in meiosis, it does, because you're going to pull a set half of the DNA one way and half of the DNA in the other direction. So it is important which chromosomes pair lines up where. And that gives rise to our second process, if you like, of creating genetic variation in what's called independent assortment of homologous chromosomes. So when we have the independent assortment of homologous chromosomes in metaphase 1, that creates genetic variation. So where they line up on the equator does really influence which chromosomes go into which cell that we're going to form at the end. So this video is just to explain meiosis, the creation of 4 haploid non-identical cells from one, designed to produce sperm and egg that have half the DNA in, so that it could form a fully formed zygote with a full set of chromosomes upon fertilization. And we've just talked about genetic variation and how it comes about in prophase 1 during something called crossing over, and in metaphase 1, where you have a random assortment or alignment of those chromosomes along the centre of the cell, called the independent assortment of homologous chromosomes. Now this video is a little bit longer, but it's because this process is quite in-depth compared to mitosis. It has that extra layer of difficulty attached to it. But I hope all that helps.